Hello, this is Chapter 4, Micro Lecture number 3. In this one, when we're looking at genetics and cellular function, we're going to take a close look at DNA and mitosis. And mitosis is just going to be how does the cell split. So when you're looking at mitosis, it's going to be the ones with the phase at the end, the prophase, the metaphase, anaphase, and telophase. But we're going to be looking at it, what does DNA happen? Or what happens to DNA during mitosis? So in this lecture, we're going to look at how is DNA replicated. How does it grow? How does it produce one strand into two? And how do we then split that strand and that genetic material during mitosis? We're going to look at the consequences of replication errors, but I think most of you understand that after going through those other lectures, DNA is just going to code for a protein. Proteins can be used in the cell membrane, can be used all over the body for all these different things. So if you don't have a working protein, it can cause many different things down the line. We're also going to look at the life history of a cell. What are the events of mitosis, the prophase, the metaphase, anaphase, telophase. I'm going to keep repeating this because repetition will help you memorize this by the end of this lecture. And then finally, we're going to explain how the timing of cell division is closely regulated. You don't want things to proceed before the other stuff that happens in there is finished. So before a cell divides, it must duplicate its DNA so it can give a complete copy of all its genes to each daughter cell. And since DNA controls all cellular function, this replication process must be extremely exact. So just some background information. DNA. It's a long parallel chain of nucleotides. Those chains are held together by hydrogen bonds. And these are going to be the hydrogen bonds between the nitrogenous bases. And there's 4 nitrogenous bases. Adenine, A, thymine, T, cytosine, C, and guanine, G. So whenever you have a T, it's going to bind to an A. If you have a C, it's always going to bind to a G. Or if you have an A, T. If you have a G, a C. So AT, CG are what you're going to be making in those pairs between all the nitrogenous bases. And those nitrogenous bases are going to be what we're going to look at now on how do we replicate those. So to give you some more background information, DNA is the genetic information stored in a sequence of base pairs. And this is going to be known as genetic code. So the A, G, C, all of that code is going to be known as your genetic code. And that genetic code codes for a specific protein. That protein then go down the line and can make many different functional things inside of a cell. It's going to be stored via a triplet code. A triplet is just a sequence of 3 nitrogenous bases. So it can be AAA, it can be CTA, it can be any code of those 4 things, or those 4 nitrogenous bases for DNA. And those are going to be known as triplets. Those triplets will then be transferred into RNA, and those RNA codes will specify a single amino acid. So how DNA makes RNA, RNA will then code for an amino acid that gets held together by a peptide bond, and that's how you have a polypeptide, which is just another fancy word for a protein of something that your body is going to make. When you look at the word gene, this is the functional unit of your heredity. So this contains all the DNA nucleotides to produce a specific protein. The size varies from 300 to 3,000 nucleotides, and this variability is some proteins are small, some proteins are big. If the protein's big, then it's going to have a lot of nucleotides. If the protein's small, it's going to have a lot smaller one. But don't just think that this is the protein and then it stops. But remember the function of the cell. The cell's going to be there to put finishing touches on it in that factory we talked about in the earlier chapters called the Golgi apparatus. So you might have a protein that becomes added with a lipid. It might be added with a carbohydrate. It might be added with a mineral like iron that can make hemoglobin. So all of these things are going to be built, but pretty much DNA is going to code your genetic. That genetic code is going to be 3 individual ones. You will probably have to learn these if you go on to higher classes like biomed and biotech, and also some of the biochemistry classes. They'll make you learn specific triplet codes, and then all you're doing is what's the gene and what does that gene code for, and it's going to be a protein. So just for example, these are some of the genetic codes in protein synthesis. You have AAA we talked about, and that can code for TTT, and then because there's no T's on DNA or on RNA, those T's get transferred to U, and that will code to phenylalanine. So all of these different codes for specific amino acids. We're not going to look at them. We're not going to kind of memorize them at this stage, but just to kind of give you an idea, I like to show you how C becomes a G, G becomes a C and a C, and that coding strand becomes the messenger RNA, which is the same in this case because the only thing that changes in RNA is T to U. And then you have the anticodon, which is the copy of that, and that is alanine. And you have a bunch of amino acids. Tryptophan is one of them, methionine, proline. All of these will then be put together via peptide bonds into a protein. So what is this law? The law of complementary base pairing. So this just means all the things we've talked about. We can predict the base sequence of the one DNA strand if we know the sequence of the other. It enables the cell to reproduce one strand based on the information in another. And this is important because if I have an A, I told you that A is always going to bind to T. C is always going to go to G. So if I know it's A, T, T, the other strand is going to be a T, A, and an A. And this is the basis of DNA replication. So let's take a look. So you have many steps in DNA replication, but first you have to unwind the helix. And you have to remember, the helix is going to be wound all around these histones, which are those little disc-like shapes. So first you need to take off the DNA helix off of those histones. Then you're going to have another enzyme called DNA helicase. And DNA helic and that ASE tells you that this is an enzyme that's going to open up the DNA helix. And it opens up one short segment of helix at a time, and this exposes its nitrogenous bases. And this causes a replication fork. This is the point at which DNA is opened up, like 2 separate halves of a zipper. And then what happens is DNA replication proceeds. The enzyme DNA helicase unwinds the double helix, creating 2 single strands. And DNA polymerase can only synthesize DNA in a 5' to 3' direction. And then this poses a problem for the lagging strand, which we'll talk about in a little bit. And this 3 to 5, these are just the number of the sugars. So we'll take a look at this later. But pretty much, you unwind it from the histones, the DNA helicase unwinds the helix, exposes the bases, and then DNA polymerase is a molecule that's going to move along each strand, and it reads the exposed bases, and then it brings in a complementary nucleotide that matches up with that one base. So again, if it's A, it's T. If it's C, it's G. If it's a G, then it's C. And it's just going to keep moving along in one direction. And yes, it's not important for this class, but 5' to 3' per direction, that's going to be the way it always is going to go. And we'll kind of look at this. I'll show you what that means, and there's a good picture coming up. So then you have the polymerase molecule moving away from the replication fork, and that's going to make a short fragments called Okazaki fragments. And these Okazaki fragments are going to be moving 5 to 3, but because they're moving away from the replication fork, because it's going to be unzippered, if you remember with the DNA helicase, these are Okazaki fragments, and then you have something called DNA ligase that's going to join them together. And this is how you make the lagging strand, which we'll look at in a little bit. And from the old parental DNA molecule, 2 new daughter DNA molecules are made, and this is called semi-conservative replication because you have a new strand that's brand new, that's based off of the older one. So each daughter strand has 1 new helix synthesized from free nucleotides and 1 old from the parental DNA. Let's take a look. So if you're wondering what 3 and 5 prime is, it's just the sugar. So these are the little sugars right here, and you can see the 1 prime is always where the base is going to be. So this is A being combined to T, and T is going to be bind to 1 prime. If you look here, 1, 2, 3, 4, 5. So 5 is where the phosphate is. And that's how you get the 5 prime and the 3 prime. It's where the phosphate is attached to. So here you have the 3 to 5. And you get that, and you get the numbers this way because it's 1, 2, 3, 4, 5. So the 5 is pointing down in this case instead of pointing up. So all of this fancy stuff that's not going to be on the test, but it is kind of interesting to know. If you can understand this, this is going to be the most difficult thing in DNA replication. So here you have the DNA helicase. The DNA helicase is going to unwind this green thing. It's going to unwind the DNA double helix like a zipper. And because this is moving in the 5 prime direction, this DNA polymerase is going to read it one at a time. So every time one gets exposed, it says, ooh, that's a T, I'm going to put a A. Ooh, that's a C, I'm going to put a G. And that's the way it's kind of going. Now, the lagging strand still has to read it in one direction. So it has to go from this to this to this to this. And you can see how once they get up to a direction, a point where it's right next to it, you have DNA ligase that has to go in there and then bind the two together. And that's kind of what it looks like, because you don't want to unwind the whole double helix molecule at a time. What you want to do is little parts at a time, and then those are going to be put together. And here's another view of it. So you still have the 5 prime and the 3 prime. 5 prime is moving in this way, and then 5 prime is going to go in this direction. And this is the Okazaki fragments that then get bound together by DNA ligase when they get close together. And that's how DNA is going to work. There's your replication fork. There's your DNA helicase, that's unwinding the helix. And then you have another one back here that winds it back together. And now you have a brand new strand of DNA. One made from the old parental DNA and one brand new daughter DNA. And that's semi-conservative replication. So if you kind of memorized all that, you got it, you kind of understand it. DNA replication is a little bit easier to understand than transcription and translation, but still watch the YouTube videos, take your time on that, memorize those terms, law of complementary base pairing. I know you've heard that term a couple times at this point, and semi-conservative replication. So a couple new terms there, but take your time. And then this is just kind of a look at if errors in mutation occurs. So if you have errors with your DNA replication, you're going to have an error in that new cell. That new cell can then produce proteins that either don't work, and if they don't work, then they're not going to be able to create that protein. If you can't produce that protein, that cell cannot produce whatever that went to. Maybe it can be a receptor on the cell. That cell will no longer be able to read a certain biochemical marker or a receptor that kind of looks for neurotransmitters. It can't be turned on by certain things. So if it doesn't have a way to fact check it and to kind of correct these errors, then the cells, the older you get, have more errors, more things fall through, and then more things fall through, the cells start to die. So there are multiple modes for correction and replication. It double checks it. So you have another protein that goes by just to make sure that each base is paired to the correct base. So A never gets bind to a G or a C. It's always A to T. So there's a proofreader that's going through there. And this results in 1 error per billion bases replicated. That still sounds like it's a lot of or some of you might even think that 1 error per billion bases, that's not much. But if you think about it, your DNA right now in your body, in multiple cell types, in multiple tissues, they're all going through mitosis right now, which means they already went through DNA synthesis. And if this happened over decades and over many years, those 1 billion base pairs replicated could form cells that do not function properly. That's usually why the older you get, the more breakdown of collagen, the more breakdown of other types of proteins, you'll see them more and more the older you get. Then you also have mutations, where if you have you're out in the sun, radiation, you can have different viruses that can come infect different types of cell types. You can work in an environment where you're dealing with chemicals. These chemicals can start breaking down DNA molecules. And these mutations just cause the DNA structures to replicate more errors. And then some mutations, no effect. Others kill the cell. If it kills the cell immediately, that's actually good, because then it can't pass on that bad code to the next cell. And that brings us to cancerous. And the cancerous cells, these start bringing on these mutations to multiple cells where they grow out of control. So, and some can also cause genetic defects in the future, in future generations. So errors and mutations, it sounds like we have it under control, but over time, eventually your DNA replication and the DNA synthesis starts to break down too. So now, the last couple of slides, we're going to look at the cell cycle. So you have 2 phases, interphase, which is in green, and then the mitotic phase, or the M phase, is going to be up top. The easiest way to think about it is these are 2 separate phases. Interphase is G1, S, G2, and then mitosis is how do we split the cells in 2. So if you think about it, interphase, you're just growing everything in the cell to make duplicate copies of everything. If you have 4 mitochondria, by the end of interphase, you're going to have 8. If you have 1 Golgi complex, at the beginning of interphase, you're going to have 2 Golgi complexes at the end. Anything that you have in the cell, at the end of interphase, you want to have 2 copies, or a duplicate of it. And in synthesis, or S, this is where DNA replication is going to occur. So it's kind of easy for this class, because in G1, accumulates material needed to replicate DNA. S, DNA replication begins. G, this is everything gets duplicated, all the enzymes that control the DNA repair, and at this stage in G2, 46 DNA molecules that were present at the beginning are now doubled to 96. And if you're like, okay, I'm not really good at math, it's not 96. It's 92. I need to change that. But it's double. So 46 to 92. And then in the M phase, this is where you're going to tear the cells apart. You're going to take everything and you're going to kind of make sure that the 1 cell will eventually become 2 cells. An interphase is a collection of G1, S, G2. Some cells don't go through mitosis, muscles, nerve cells that kind of go through rest, and the cell duration varies between the different cell types. When you're looking at the cell life cycle, what's important, there's two different types of cell division. Mitosis is the one we're talking about. And mitosis is when you make two different daughter cells. And each of them are going to have 46 chromosomes. Or they're going to have 23 pairs of DNA. Or 23 pairs of chromosomes. So by reading this very closely and say 46 chromosomes, or they're 23 pairs, because pairs is double, mitosis is going to make two complete functional cells. Meiosis, we look at the end of this of AMP2, this is where you're going to have sex cells. We're going to have reproduction of the sperm and the egg, and this is where you're going to cut down the genetic material in half. So instead of having 46 in a normal cell, you're going to have 23. And 23, you can't have, 12 point or 11 and a half pairs. So 23 chromosomes is just, you take everything in half and you get some of the genes or the chromosomes from your mom, some of them from your dad. And that's going to be meiosis. That's at the end of AMP2. But mitosis is what we're going to look at. So this is cell division is going to be in the bodies except for the eggs and the sperm, which I mentioned because that's meiosis. And then the function is you're going to have one fertilized egg, and those one fertilized eggs are going to grow into around 40 trillion different types of cells. So from one egg, you can have many different types of skin cells. Those skin cells are part of the 40 trillion, but they all come from the one egg. You can also have liver. You can have all of the cells in your body all come from one egg, and they're all functions of mitosis. So everything after birth is going to be coming from that one different cell. That's why your genes don't change throughout your life. The genes you're born with, the 23 from your mom, the 23 from your dad, they're going to keep growing throughout your entire life. But proteins, enzymes, environmental factors kind of guide them into what they're going to grow into. We're not looking at that in this lecture. All we're going to look at, what are the four basic phases of mitosis? Prophase, metaphase, anaphase, and telophase. And if you look at it, mitosis is all going to have phase at the end of it. The three phases of interphase are G1, S, G2. Those are letters. So you shouldn't get mitosis and interphase mixed up. So I'm going to go over everything on this slide, and then I'm just going to have the other slides remaining in this lecture just to kind of make some points. But when you're doing prophase, the nuclear envelope is going to dissolve. The nuclear envelope is going to dissolve because you want those chromosomes, you want that DNA that's locked in them to start to come out. And you're going to have these centrioles that are involved in cell division. This is cell division. So those centrioles are going to start hooking on to the chromosomes and start guiding them to either the top or the bottom of the cell. All of that's happening in prophase. Then metaphase is when everything lines up on the metaphyseal plate. Or, much easier, metaphase starts with the letter M, so does middle, so everything's lined up in the middle. Middle, metaphase. Then, if you can draw a line between the chromosomes or DNA on top and the bottom, that comes after metaphase, and the one that comes after anaphase is anaphase. So anaphase is when things are going to be going to the one side or the other side. Then when you have the nuclear envelope start to reform, it's telophase. And if you look at it, P, M, A, T for PMAT, prophase, metaphase, anaphase, telophase. And this is just everything I kind of looked at. So if you see the nuclear envelope dissolve, that's going to be the beginning of prophase. And when you see the chromosomes just kind of all wound up in the center of the cell, that's going to be a classic clue of prophase. Metaphase is when chromosomes line up on the equator or the metaphyseal plate or metaphyseal line. And then anaphase, you're going to have enzymes that cleave the two sister cells apart. That's going to be the beginning of cytokinesis. And you're also going to have them moving up the spindle fiber towards along the spindle fibers to the centrioles itself, and this is when you're going to be able to draw a line between the chromosomes on top and the chromosomes on the bottom. And then finally, the dead giveaway that telophase is happening is when you have the nuclear envelope reform. And at the end of that reforming of the nuclear envelope, at the end of telophase, you'll have two daughter cells. And I kind of mentioned this term, cytokinesis. Cytokinesis is the pinching of the cells. So you'll see the beginning of cytokinesis in anaphase. And after telophase gets done, that's when you have cytokinesis finishing up. Because cytokinesis is that pinching, and that pinching will then allow the two cells to pull apart. And then finally, the timing of cell division. And this is where we end. The cells divide when they have enough cytoplasm for two daughter cells. They also replicated their DNA. Because without DNA, there's no point to produce a daughter cell. They have adequate supply of nutrients. They're stimulated by growth factors. There can be chemical signals secreted by blood platelets, kidney cells, and other sources. Neighboring cells can die, which opening up spaces for new cells. That's what happens in nervous and muscular tissue in most of those cases. And they start to divide and they stop dividing when they're too close to their neighboring cells. There's no nutrients or growth factors for them to actually signal to grow. Or they undergo contact inhibition, and this is the stopping of cell division in response to contact with other cells. So if you don't have any room to grow, they don't grow. But if you think about what happens in cancer, one, they don't die. They lose the ability to stop growing, and they keep growing uncontrolled even if they don't have any room. So you can see with mutation, all of this comes back together where if you can't control the cell division, it can lead to cancer where cells start to grow into areas where they're not supposed to, which will cause a lot of trouble. And that concludes this lecture. Good luck.